part 2. Alright, listen. This concept is very important for your upcoming MST 2 exams. MST 1. MST 1. I was taking a slow approach when I am delivering my content in the theory classes, and I was lagged behind one particular topic in the syllabus called toxic granulation. Toxic granulation, and I also need to complete one topic called lymphoproliferative leukemias. Lymphoid leukemias, that I will finish tomorrow. Meanwhile, I need to cover this topic and I will cover today. This you will get 5 marks and 1 2 marks, or you might also get a 1 10 mark. I don't know which question you will get in the question paper. So from this topic you will get damn sure you will get some 5 marks from this topic. So everyone pay utmost attention, okay? Listen. You need to understand what are inclusion bodies. What are inclusion bodies means? Can anyone tell me?
They are the storage.
Inclusion bodies. Listen, inclusion bodies means these are some abnormal proteins or some abnormal structures, components. Okay? Some abnormal substances or components present inside the cell. Present inside a cell. Okay? Maybe some abnormal components present inside a cell, which normally won't present. Okay? Such components are called as inclusion bodies. The presence of inclusion bodies, I will give few examples. Okay? Examples of inclusion bodies. One is Hens bodies. Hens bodies we have seen in case of
G6PD.
what disorder?
G6PD.
G6PD. In G6PD what happened? G6PD. In G6PD what happened? Red cell membrane became fragile. That fragile membrane started breaking and those pieces of membrane suspended in a red blood cell, leading to appearance of an inclusion body, that is Hens bodies. Is it clear? Second condition. Reticulin fibers. Some reticulin fibers can be seen. Reticulin fibers can be seen in normal condition also. In what cell we will see reticulin fibers? In reticulocyte. That means immature RBC. That means the presence of inclusion bodies indicates some problem or infection or some condition in the patient body. When patient has an anemia, when there is a severe RBC breakdown, what happens to the bone marrow? Listen, there is an anemia. Anemia means reduced RBC count because of increased RBC destruction. Some malaria, something destroying the RBC, leading to anemia. During this anemia, what happened? Bone marrow started producing more red blood cells into the circulation. A side effect of increased bone marrow activity results in increased reticulocyte count. So, presence of increased reticulocyte count indicates appropriate bone marrow response to anemia. So, we can say that reticulocytes is reticulin is a type of inclusion body. Detection of inclusion bodies indicates some issue in the body or condition in the patient. Is it clear? That is how we are using inclusion bodies. I will give few more examples. Another example will be in case of rabies infection. In case of rabies infection, if I take the patient cheek cells, okay, if I take the patient cells, in the cells I will see a specific inclusion body called as Negri bodies. Negri bodies. So very simple. You are walking on this.
Fatima is there. Fatima is there.
Yes, I got a patient who was got a dog bite. One dog bited a patient. Okay. Now I need to know whether my patient developed rabies or not. Chances are there. From dog we will get rabies. How I will know? If the rabies virus entered into the patient, this virus will interfere with the cells, and because of its interference, I will see negative bodies. So I will take patient cells and I will detect negative bodies. If negative bodies are there, he catch an infection. If there is no negative bodies, I cannot give that injections, rabies injections. Okay. So see, simple blood smear test, simple cell examination can give me a viral status in the body. Okay. Another very interesting inclusion body, I like it a lot. That is, listen, coming to the female genetics, XXXY. You know, right? Listen, have you any time observed X and Y especially? The X chromosome looks very large, but Y chromosome looks very small. Do you know?
Yes, sir.
Actually, the X is a dominant allele. Actually, the entire human anatomy is developed by X chromosome. Complete anatomy was developed by X chromosome. The only reason what Y doing is, Y will suppress some of the X activities. Y is only there for suppressing some of the X activities, that's it. That's why I said in my last lecture, we all born as females first. Okay? That's why even males will have nipples but without chest enlargement. Okay? Because we are designed to be a woman first. Okay? This is really interesting thing at genetic level. Okay? Now listen, very important. Coming to the females, only one X is sufficient for human anatomy, I said. Then we got two X's. That means we are superhumans. That is not good. What the nature did is, nature will inactivate one of the X chromosome in females. Okay, in all females, genetically they are XX, but they are running on only one X. Got my point? In all the female cells, that inactivated X, okay, I will write small x, this is inactivated X. This inactivated X will be present as a small body within the nucleus. This body is called as bar body. This body is called as bar body. Okay, bar bodies are arised due to inactivation of one X. Inactivation of one X lead to a bar body. So technically, where we will identify bar bodies? Only in women. I cannot see bar bodies in males. Okay, because they only have one X and that is running. So I cannot say inactivated X bar body. But in women, I can see a bar body. Is it clear? That means, even if I don't see gender on the blood sample, I can still identify whether it's a male sample or female sample with a bar body detection. Bar body can be seen in almost all white blood cells in the women, not in the males. Is it clear? Yes.
What happens in other sexes?
What happens in, yeah. Now listen. I said that trisomy issues. Disjunction. I got a male. I know this is a male blood sample, but in the male blood, I detected a bar body. How is it possible? How a male got a bar body? He might have a XXY condition, that is skin filter syndrome. Okay, the third type of humans. Genders, the third genders, yeah.
Sir, XO.
XO is also there, and also don't think that women are immune to these issues. Even there are super females with triple X condition. Okay, we will see two bar bodies in these women. Unfortunately, these individuals are sterile. They can't have children because they
Turner syndrome.
Turner's.
XO.
Yeah, Turner syndrome. Turner syndrome, then Down syndrome. There are multiple syndromes. Okay. See, now we are discussing the beauty of inclusion bodies to detect the genders and the third gender also. See, so that is the beauty of inclusion bodies. So now you understood the significance of inclusion bodies with these examples. Am I correct? Any confusions?
No,
No, right? Now, the third phase of this lecture, that is the important part. Tell me the types of white blood cells. We have granulocytes and agranulocytes. Tell me the types of granulocytes.
Neutrophil.
Neutrophils, basophils, eosinophils. Then
T and
monocytes and lymphocytes. What are the most prominent cells? Tell me in sequence. Never let monkey eat banana. 60, 40, 8, 4, 0 to 1. See, everyone knows these things. Haven't you seen some interesting pattern here? What is the most abundant cells here? Neutrophils. That means it is neutrophils who are doing the leading, immune function. It is neutrophils who are doing the leading protection function. Yes, Miss Rejoice. Anything interesting or funny there? Yes. No, she's still bluffing here. Blushing. Anyway, see here. Yeah. That's why neutrophils are called as first line of defense cells. Okay. Usually neutrophils are very good at bacterial infection. Okay. When any infection entered into the body, it is the neutrophils who will attack them. Is it clear? Now, let me tell you one interesting thing. Unfortunately, you got a food poison. You got a Clostridium botulinum bacteria into your circulation. There is a heavy load of bacterial infection in the circulation. Now what happens? Rather than 60% of neutrophils, now we need the demand of neutrophils increased. Maybe there will be 110% of neutrophils in the circulation. The neutrophil count might increase during an ongoing infection. Is it clear? How more neutrophils came into circulation? Because our bone marrow sent more neutrophils into the circulation. Okay? In the bone marrow, we have a stem cell, hematopoietic stem cell. This stem cell, okay, there is a signal from the outside environment. That signal will be passed to the bone cell stem cells, and those signal molecules are called as cytokines. Okay? Listen. Imagine you as a stem cell and you want to convert to many types of people, okay, many types of cells. Either neutral, isno or beso. How you will decide? You are deciding by the input. The cytokine cell differentiate into a neutrophil. So the stem cell started dividing into a neutrophil. It's converted into a neutrophil. Very beauty things happens here, okay? Gene switches. Every cell, every stem cell can be able to convert to any cell in the body. But the other genes will be inactivated, only the neutrophil genes will be activated. How these switches are working in genes, that is very fascinating. Anyway, the gene switches we'll see in any other class. Up to here is clear, right?
Yes.
Stem cell converted into neutrophil. Just like during a stress, there is a chance of increased reticulocytes. Is there a chance of increased abnormal neutrophils in case of infection? There is a chance of, arising of some abnormal neutrophils into the circulation. There is a chance, right? Yeah. These abnormal neutrophils, if they are more, okay, one reason is bone marrow response abnormal neutrophils. So these are good abnormal cells, friendly abnormal cells. Second one, now my ongoing virus or bacteria destroying the neutrophil in many types. Okay, so now I have two types of neutrophils. Neutrophil under attack, neutrophils under normal response. Now see, I will show various types of neutrophils here. A bacteria attack, okay, the neutrophil, yes. Listen, there will be a hyper nucleus. The nuclear content will be more in case of infection. So hyperpigmentation can be seen. Hyperpigmentation can be seen. Okay. Another one, ongoing bacteria destroyed. If bacteria destroyed, this looks in one type of cell. If virus destroyed, that looks like this. If other condition, okay. If you take any toxins, your neutrophil looks in another condition. Just by seeing the neutrophil, the inclusion bodies inside the neutrophil, we can guess what's happening to the patient. Just by seeing the peripheral blood smear, by seeing the anatomy of the neutrophils, I can able to guess the condition of the patient. That is why detection of neutrophils has a significance. Is it clear?
Yes, sir.
Now, many scientists connected many inclusion bodies to many disorders, and they have given those scientist names to various types of neutrophils. Now, I will tell some interesting things, these things, okay? Some patients are genetically, they will get a weak or abnormal neutrophils. Genetically, they have issue with neutrophil. That means their neutrophils are not good at attacking bacteria. So these patients will catch very frequent infections. Or I will give you a very basic example. If you see in the movie I, okay? Everywhere from birth to death, they will have infections on their body. And they cannot recover from that infections because their neutrophils have issues. Till death, they will have some infections and they will die due to some sepsis only. Okay, these are very painful conditions, no treatment because they are genetic. Only we can, sustain the life by giving some antibiotics frequently. Okay, you just imagine those consequences. Okay, if this condition became so severe and this neutrophil started migrating to whole body, you will get systemic infections. And patient might die due to this systemic infection. Whole body they will have sepsis. Okay, such diseases are called as toxic granulosis. I wrote here, right? Toxic granulosis disorders. There are various types of toxic granulosis disorders. What are the toxic granulosis? These are the granulocytes and they have some toxic conditions. Some granulocytes have large vacuoles, okay, lysosome vacuoles. Some have granular inclusion bodies. Some have various abnormalities, leading to various problems. Okay, on board it looks nothing, but if you see the patients, you will understand. Pathetic, okay.
Sir,
Yeah.
and
More, okay. This can happen both genetic and epigenetic. That means virus, bacterial attacks also can lead to these problems.
sir, any condition?
Some viruses. Huh?
Any condition example, sir?
Now I will give you the examples. Write down the toxic granulosis. I will give 4 disorders. The 4 disorders. First one,
मैं नोट ही नहीं करेंगे.
Higashi syndrome. 1. Higashi syndrome. Okay, this is called as Higashi syndrome. CHS. Higashi syndrome. Okay, write down points. Only 2 to 3 points is sufficient. First point: abnormal lysozymes in the neutrophils. Abnormal lysozymes in the neutrophil. Giant granules in the neutrophil.
Sir, first one,
Abnormal lysozymes in the neutrophil. Giant granules in the neutrophil. Giant granules, large granules can be seen in the neutrophils. Impaired microtubules can be seen in the neutrophils. Impaired microtubules. Okay, microtubules. Do you remember cytoskeleton of the cell?
Yes, sir.
The cell shape was given by microtubules. They are the fabric of cells, the scaffolding of the cells. Okay. Yeah. Symptoms. What happens to the patient? Symptoms. Okay. Symptoms are severe immunodeficiency. Severe immunodeficiency. Severe immunodeficiency. Second, partial oculocutaneous albinism. Partial oculocutaneous albinism. Oculocutaneous. Oculo, eye cutaneous skin. Albinism. Loss of skin pigmentation. Albinism means, have you seen some patients, some people outside, they won't have color. They will be bleached, they will be very white. Okay, genetically problematic patients. Not patients, people. Right? Abnormally white people. Actually, they have issue with neutrophils. See, even neutrophils and melanocytes have connection because they have same stem cell lineages. Skin pigmentation is given by melanocytes. Melanocytes are a subset of same stem cells actually. See, study of stem cells has good things. Okay, this is the second symptom. Third symptom, albinism can be seen. Third one, neurological problems can be seen. Neurological problems will be exhibited. Neurological problems will be exhibited. Third one, bleeding disorders can be seen in the patient. Bleeding disorders can be seen in the patient. Okay? I will give you the pictures of Higashi syndrome neutrophils in the classroom, okay, in the group. So this is all about Higashi syndrome. Higashi syndrome clear, right?
Yes, sir.
It's an immune deficiency where you will see lysozymes and large vacuoles, large granules, giant cells in the neutrophil. Higashi done. Then second syndrome, yes should be. Yes. Second one. Toxic granulation. Toxic granulation condition. Toxic granulation condition. By the name itself, you can understand toxic granulation condition. Write down the definition. There will be a hyperpigmented, large number of porous granules. Hyperpigmented, large number of porous granules. Chorus, that means rough. Chorus granules present in a neutrophil. Chorus granules present in a neutrophil. I will conduct quiz, okay? Don't escape, don't think. Okay, yes. Hyperpigmented chorus granules present in neutrophils. Clear, right? Yeah. Symptoms. Prone to severe bacterial infections. Patient is prone to or susceptible to severe bacterial infections. Patient is susceptible to severe bacterial infections. Severe bacterial infections. This is also seen in inflammatory conditions in the body. These cells also seen in inflammatory conditions in the body. And these cells will indicate the severity of the infection in the body. These cells also indicates the severity of the infection in the body. The severity of the infection in the body. You might ask me a question, why only neutrophils? What about other cells? Even other cell abnormalities are there, but that will be beyond the scope of this subject. Okay? At least we will understand neutrophils. That is it. Okay?
Sir, last point.
Severity of the condition. Third one, severity of the condition can be assessed by understanding these cells. Yeah. The severity of the bacterial condition. Okay. Next, third one, third disease. May-Hegglin anomaly. May-Hegglin anomaly. Okay, anomaly. I guess A-N-A-M-O-L-Y. Anomaly. May-Hegglin anomaly. In this, dull bodies are detected. Dull bodies. Okay. D-O-H-L. Like this. Dull bodies. Some French language. Okay. Some French terms. French or Latin. Dull bodies will be detected in May-Hegglin anomaly. Okay. Round bluish inclusions will be present in the cytoplasm of neutrophils. Round bluish inclusion bodies will be detected. Round bluish inclusion bodies will be detected in the cytoplasm of the neutrophils. In the cytoplasm of the neutrophils. These cells are seen in inflammatory conditions, trauma, burns, skin burns, and heavy stress to the bone marrow. CBC findings include, next point, CBC findings include complete blood count, okay? CBC findings include severe thrombocytopenia, that means reduced platelet count. Reduced platelet count, giant platelets, presence of giant platelets will be very large. Presence of giant platelets is characteristic features. are characteristic features. Last and final, fourth disease, fourth disorder.
Sir, wait a minute.
Fourth disorder. Okay. Fourth disorder, write down. Pegler-Hewitt's anomaly. Pegler-Hewitt. Okay. Pegler. Okay. Wait. Pegler. P-E-G-L-E-R. Pegler-Hewitt. H-U again something E-T. Hewitt anomaly.
So P-E-L.
Anomaly. Okay. Pegler-Hewitt anomaly. Pegler-Hewitt anomaly. Okay. These are some Latin scientists. Okay. Their names.
Sir, one pad low.
If Keshav detects something, Keshav anomaly, you know, that's how these cells came here. Yeah, so they get the scientist names. Okay, Pegler-Hewitt anomaly. Okay, listen here. Write down. Hyposegmentation. Hyposegmented neutrophils can be seen. Hyposegmented, not hyper. This is hypo. That means less. Pale colored neutrophils. Okay, hyposegmented. Neutrophils can be observed, can be seen or observed. Okay. Next, usually asymptomatic and less risk to the individuals. Usually asymptomatic and less risk to the individuals. Asymptomatic, that means it won't cause much issues. Asymptomatic and less risks. Keep in mind why we said asymptomatic, because when we do the DLC, in DLC it looks like pale neutrophils. We will think we haven't added dye issues. We will think dye issue. So that's how it will be asymptomatic. Anyway, another important thing.
Hyposegmented neutrophil.
In rare cases, these patients are likely to develop. In rare cases, these patients are likely to develop myeloproliferative leukemias. See, that means we can't even. Patients will develop leukemia, blood cancer. Leukemia. What type of leukemia? Neutrophils derived from myeloid lineage. So myeloproliferative leukemia. Myeloproliferative leukemia. Okay. Now write down one over. Okay. Myeloproliferative leukemia. Okay. Last and final point I didn't forgot to discuss. May-Hegglin and Pegler-Hewitt disorders are a very rare genetic disorders. Both of them are very rare genetic disorders. Very rare genetic disorders. Very rare genetic disorders. Okay. Again, self-boasting my subject. A simple hematological studies giving a deeper insights into patient problems. No other branch in medicine can give as much insights as hematology. Even microbiology relied on hematology. Even biochemistry relied on hematology. It all starts with a simple CBC. You just see whether there are normal values, RBC, WBC, platelets. If you identify some issue, definitely there are root causes of other issues. Okay. So this is the beauty of hematology. Okay, you must be experts in hematology first of all.